So today we will learn the Chapter 7, RNA-directed DNA methylation in plants. Chapter 7 is composed of the 5 subjects, started from the RdDM in plants, and also we will learn the DNA methylation in plants. And after that we will learn the specific mechanism in the RdDM. So finally we will learn the paramutation. In this slide, I will explain the first subject, RNA-directed DNA methylation, RdDM. So actually, initially, the RdDM is the epigenetic process first discovered in the plant, and also the RdDM is the host defense mechanism against the virus or transposon in plant. So this is the one example. So this is the tobacco leaf. This is an uninfected leaf. So this is the tobacco leaf, which was the TMB virus infected leaf. Okay. So after TMB virus is impacted the leaf plant, so there are host defense mechanism happens. For example, this is the viral double-stranded DNA. So initially, after the virus impacted into the host, the plant, the viral RNA can be transcribed. Okay, so after the viral RNA is transcribed, host defense mechanism happens. So the RdDM happens. So using the RdDM, so they will make the double-stranded RNA. So, but I will not cover in detail. Okay, but after they make the double-stranded RNA, so another RdDM happens. So finally, they will make the methylation in the viral double-stranded DNA. So in this case, this viral double-stranded DNA cannot be transcribed. So this is the host defense mechanism against the virus. How about the host defense mechanism against the transposon? Okay, this is one example. So plant has the loss of the active transposon elements. So usually the transposon element is transcribed after this gene is inserted into the randomly inserted our gene of interest. So when the transposon element is inserted into the gene, so gene become the inactive, or they will become the mutant form. But in this case, so in this case, when host defense mechanism happens against the transposon. So in this case, RdDM happens. So this is a transposon elements. So RdDM make the methylation is here. So it means that transposon element cannot be transcribed. Okay, so in this case, our gene of interest can be transcribed. So they will make the active gene status like this. So this is also another host defense mechanism against transposon. So RdDM is found in the heterochromatin region. So they usually found in the heterochromatin region. However, this RdDM mechanism is not found in heterochromatin region around centromere. So specifically in the plant, in the centromere, there are different mechanisms happen. Okay, but I will not cover in details. So once again, RdDM is found in heterochromatin region. Okay, but RdDM is not found in the heterochromatin region around the centromere. Okay. In this slide, I will explain the special characteristics in the RdDM. So RdDM needs plant-specific RNA polymerase to synthesized RNA transcript. Okay, in our previous slide, we also learned that heterochromatin formation by RNAi. So at that time, RNA polymerase II involved in this step. So this is the RNA polymerase II. However, in plant, especially in RdDM needs plant-specific RNA polymerase. For example, here, this is the RNA polymerase 4. Okay, so RNA polymerase 4 synthesized RNA transcript is like this, and this is the RNA polymerase 5. So, but however, the difference between RNA polymerase 4 and RNA polymerase 5 is that RNA polymerase 5 can synthesize RNA transcript that does not have poly A. So we will see some detailed information in the following slide, okay? In this slide, I will explain the second subject, DNA methylation in plants. So DNA methylation in plants occurs in CG dinucleotide. Okay, so usually DNA methylation in plants occurs in CG dinucleotide. 55% occurs in CG dinucleotide. Okay, so usually the CpG islands contain a large number of CG dinucleotide repeats. So cytosine base is followed by the guanine base. So usually the cytosine in such an arrangement tend to be methylated. Okay, and also DNA methylation in plants also occurs in CHG. For example, this is the CHG or CHH. Okay, CHH site. So where H can be A or T or C. So CHG can be CAG or CTG or CCG is like this. Okay. So 23% of cases occurs in CHG and 22% cases occurs in CHH site. Okay. So in this slide and next slide, we will learn the DNA methyltransferase in plants. So in the previously we learned that classical maintenance DNA methylation model. There are two models available. One is conservative DNA methylation. So conservative DNA methylation means that recreates methylation pattern on hemimethylated DNA. So at that time, the DNMT1 is involved in the conservative DNA methylation model. So here in plants, MET1 and the CMT2 or CMT3 is involved in the conservative DNA methylation model. Okay, so let's start from the MET1. So MET1 is the also DNA methyltransferase in plant. So actually this is the plant DNMT1. So plant DNMT1, we call it as MET1. So actually this MET1 is the CG methyltransferase. However, CMT2 and 3, so this CMT2 and 3 is also involved in the conservative DNA methylation. So however, CMT2 or CMT3 is the CHG methyltransferase. Okay, the difference between the MET1 and CMT2 or 3 is that MET1 is CG methyltransferase. However, CMT2 or 3 is the CHG methyltransferase. Okay, so the function of MET1 or the function of CMT2 or 3 is the recreate methylation pattern on the hemimethylate DNA. So recreate the methylation pattern on the hemimethylate DNA. However, this MET1 is not related to RdDM, so RNA-directed DNA methylation, but this CMT2 or CMT3 is related to RNA-directed DNA methylation. Okay, let's summarize here once again. So in conservative DNA methylation model, okay, so in plant, especially the MET1 and CMT2 or 3 is involved in conservative DNA methylation. The difference between the MET1 and the CMT2 or 3 is that MET1 is CG methyltransferase and also CMT2 or 3 is CHG methyltransferase. So, and also MET1 is not related to RdDM, but CMT2 or 3 is related to RdDM. So in this slide, I will explain the DRM2, another methyltransferase. So in our previous chapter, we learned that de novo DNA methylation. So this is a de novo DNA methylation. So this de novo DNA methylation means that newly synthesized DNA methylation. So there was no DNA methylation in both strands. So after the de novo DNA methylation happens, both strands can be DNA methylated. So this is recorded as a de novo DNA methylation. So at that time we learned that DNMT3A or DNMT3B is involved in de novo DNA methylation. However, in plants, DRM2 is involved in de novo DNA methylation. Okay, so DRM2 is the function as the plant DNMT3A or DNMT3B. The main function is de novo DNA methylation. So de novo DNA methylation happens in CG dinucleotide or CHG sequence or CHH sequence. Okay, and also DRM2, the de novo DNA methylation is highly related to RdDM. So we will see the detailed function of DRM2 in the next slide. Okay. So in this slide, I will explain the third subject, the molecular mechanism of RdDM. Okay, in RdDM, there are two types of RdDM is there. First type is the canonical RdDM. In Korean, we call it as the 전형적 RdDM. And there is also another RdDM, we call it as atypical RdDM. In Korean, we call it as a 비전형적 RdDM. So now we will focus on the canonical RdDM first. Okay, so canonical RdDM is composed of the three steps. Step 1, step 2, step 3. Now we will focus on step 1. Step 1, we call it as polymerase 4 dependent siRNA biogenesis. So polymerase 4 dependent siRNA biogenesis. In previously we learned that RDRC, so this is the RDRC. So RDRC is a synthesized double-stranded RNA because the RDRC has the RDP1. So RDP1 is RNA-dependent RNA polymerase. So after that, RDRC make the double-stranded RNA, DICE1. So DICE1 is cleavage, so double-stranded RNA, so they will make the siRNA is like this. How about in canonical RdDM? Okay, so in canonical RdDM is the RNA polymerase 4 synthesize the RNA transcript. Okay, at that RDR2. The full name of RDR2 is that RNA-dependent RNA polymerase 2. So this is based on the RNA, so they will make the double-stranded RNA. So RDR2 synthesize the double-stranded RNA. So after that, DCL3. The full name is dice-like 3. So DCL3 cleaves the double-stranded RNA into siRNA, small interfering RNA. siRNA is like this. Okay, so step 1 is that once again, polymerase 4 synthesized RNA transcriptor. After that, RDR2 bind here, and RDR2 synthesized double-stranded RNA. After that, DCL3 is cleaves the dsRNA into the siRNA. So after that, HAN1 induces 3' methylation at both ends of siRNA. So previously we also learned that HAN1. So in piRNA and PTGS we learned that HAN1 induces 3' methylation. So here is also same. HAN1 induces 3' methylation at both ends of siRNA. Okay. After that, ARG4 binds to the double-stranded RNA, double-stranded siRNA, and cleaves it into single-stranded siRNA. So after that, ARG4 and single-stranded siRNA complex is synthesized like this. Okay, so this is the step 1 procedure. So we call it as polymerase 4 dependent siRNA biogenesis. So siRNA is synthesized. That DCL3 is cleaves it into the siRNA. After that, HAN1 induces 3' methylation at both ends, and finally the ARG4 and single-stranded siRNA complex is synthesized. Okay, so this is step 1. So in this step, I will explain the second step in canonical RdDM. So we call it as RNA polymerase 5 mediated de novo methylation. RNA polymerase 5 mediated de novo methylation. So initially, the RNA polymerase 5 synthesized the RNA transcriptor like this. Okay, after that, ARG4 with a single-stranded siRNA bind to the nascent RNA. So nascent RNA means that newly synthesized RNA transcriptor. So ARG4 with single-stranded siRNA binds to nascent RNA from polymerase 5. Okay, so after that, ARG4 single-stranded siRNA and nascent RNA complex recruit DRM2. So this is DRM2. So after that, the main function of DRM2 is that it functions as plant DNMT3A or 3B. So their main function is de novo DNA methylation in CZ dinucleotide or CHG or CHH sequence. So okay, after that DRM did de novo methylation here and de novo methylation here. So this is the second step in canonical RdDM. We call it as the RNA polymerase 5 mediated de novo methylation. So in this slide, I will explain the final step in the canonical RdDMs. So we call it as this step as chromatin alteration step. So this is chromatin alteration step. So in the final step, IDN2 IDP complex, the blue complex, is bind to the RNA transcript and the 3-SNF complex together. Okay, so this binding is inhibits 3-SNF complex activity. So main function of 3-SNF, so this is 3-SNF proteins. After it bind to the chromatin structure, so it will lose the chromatin structure. However, this binding inhibits 3-SNF activity, finally prevents the step to loosen the chromatin structure. So after that, SUVH4 or SUVH5 or SUVH6 is recognized methylation and decodes information of methylated histone. So it means that after that it will recruit HDAC. HDAC6 means that histone deacetylase. So histone deacetylase is remove histone acetylation. So finally it will induce packing of chromatin structure. So this is the final step in the canonical RdDM, starting from the IDN2 and IDP complex. Finally the HDAC6 removes histone acetylation, and finally it will induces packing of chromatin structure. So in this slide, I will explain the atypical RdDM. Atypical RdDM is also composed of 3 steps. So in this slide and following slide, we will focus on step 1. Okay, in the previous slide, we learned that canonical RdDM, especially in step 1, during the step 1, RNA polymerase 4 synthesized RNA transcriptor. At that, RDL2, we call it as RNA-dependent RNA polymerase 2 synthesized double-stranded RNA. At that, DCL3 cleaves the double-stranded RNA into the siRNA. How about here? At atypical RdDMs, polymerase 2 synthesized the RNA transcriptor. So at that, RDR6, previously in canonical RdDM, RDR2 make the double-stranded RNA, but here is RDR6 synthesized the double-stranded RNA. After that, in the canonical step, is DCL3 cleaves double-stranded RNA into the siRNA. So in the atypical RdDM, DCL2 and DCL4 cleaves the double-stranded RNA into the siRNA. Especially the DCL2 cleaves the double-stranded siRNA into 22 nucleotide siRNA, and also DCL4 cleaves the double-stranded RNA into 21 nucleotide siRNAs. Okay, so this step is also the initiation step 1 step. So in canonical RdDM, so HAN1 is induced 3' methylation at both sides of the siRNA. After that, R group 4 binds to the double-stranded siRNA and cleaves it into single-stranded siRNA. After that, synthesis of the R group 4 and the single-stranded siRNA complex happens like this. Okay, how about in a typical RdDM? So in a typical RdDM, R group 2 bind to the double-stranded siRNA and also they cleaves it into the single-stranded siRNA. After that, they will synthesize the R group 2 single-stranded siRNA complex. So in canonical RdDM step, so HAN1 induces the 3' methylation at both ends of siRNA, but in a typical RdDM, there is no HAN1 the procedure. So only the R group 2 bind to the double-stranded siRNA and cleaves it into single-stranded siRNA. After that, R group 2 and single-stranded siRNA complex is synthesized. So this is step 1. We call it as the initiation step. So in this slide, I will explain the second step in a typical RdDM. So we call it as RNA polymerase 2 and RDR6 dependent RdDM. So now in this slide, I will explain the RdDM step. Okay, so polymerase 5, RNA polymerase 5 synthesize the RNA transcriptor. After that, in previous step, we found that ROG2 and single-stranded siRNA complex is synthesized. Okay. After that, ROG2 and single-stranded siRNA is bind to the nascent RNA from polymerase 5. So this is ROG2 and the single-stranded siRNA bound to the nascent. Nascent means the newly synthesized RNA transcriptor from polymerase 5. Okay, let me some details in the canonical RdDM in step 2. We also have similar step, but we will see some details. In the canonical RdDM, so we call it as the polymerase 5 mediated de novo methylation. So at that time, polymerase 5 synthesized the RNA transcriptor. After that, ROG4 and single-stranded siRNA is bound to the nascent RNA from polymerase 5. So after that, ROG4 single-stranded siRNA and the nascent RNA complex recruit DRM2. So DRM2 induced de novo methylation in the CG or CHG and CHH sequence. How about in the atypical RdDM in step 2? Okay, so in step 2 is the once again polymerase 5 the synthesized RNA transcriptor. Step that ROG2 with single-stranded siRNA bound to the nascent RNA from polymerase 5. Okay, after that, ROG2 and the single-stranded siRNA and the nascent RNA complex recruit DRM2. Okay, so after that, DRM2 did de novo methylation, especially in the CG and CHG and CHH. After that, so this DNA strand is become the methylated, so we call it as RdDM. So okay, so in this region, so this region is methylated. So in this region, so for example, RNA polymerase 2 cannot bind to this region. So it means that it can induce transcriptional gene silencing, because the RNA polymerase 2 cannot bind to the here, so they will not initiate the transcription. So that's why we call it as establish transcriptional gene silencing. Okay, so this is the step 2, and previously we also learned the step 2 in canonical RdDM. So they are nearly similar but a little bit different. For example, in canonical RdDM, it's ROG4 and single-stranded siRNA. However, in the atypical RdDM, ROG2. So ROG2 with a single-stranded siRNA bound to the nascent RNA from polymerase 5. Okay. So in this slide, I will explain the final step in the atypical RdDM. So we call it as the RNA polymerase 4 and the RDR2 dependent RdDM. Okay, so previously in atypical RdDMs, in step 1, initiation step, so usually the RNA polymerase 2 bind to the DNA region. Okay, but however, in this region, in this DNA region, RNA polymerase 5 is involved, and after that, DRM2 is methylated to this region. However, RNA polymerase 4 can bind to the methylated DNA. So this RNA polymerase 4 especially can bind to the methylated DNA. However, RNA polymerase 2 cannot bind to the methylated DNA, but RNA polymerase 4 can bind to the methylated DNA and can synthesize the RNA transcriptor. Okay, so however, so in the initiation step, we can go over it once again. RNA polymerase 2 can synthesize the RNA transcriptor. After that, RDR6 is a synthesized double-stranded RNA. After that, DCL2 and DCL4 cleaves the dsRNA into the siRNA. So in step 3, in the atypical RdDM, so RNA polymerase 4 is bind to the methylated DNA and can synthesize the RNA transcriptor. After that, RDR2. So RDR2 is RNA-dependent RNA polymerase 2 can bind to the RNA transcriptor. After that, they can synthesize the dsRNA. So after that, DCL3. So in this step 1, DCL2 and DCL4 cleaves dsRNA, but in this case, step 3, DCL3 is cleaves the dsRNA into secondary siRNA. So step 1, they made the first siRNA, and step 3, they made the secondary siRNA. That ROG4 bind to the secondary siRNA and cleaves it into single-stranded siRNA. How about in the step 1, initiation step in atypical RdDM? At that time, so ROG2 bind to the DS double-stranded siRNA. So this is a primary siRNA and cleaves it into the single-stranded siRNA. After that, ROG2 single-stranded siRNA complex is synthesized. Okay, but in step 3, ROG4 bind to the secondary siRNA and cleaves it into single-stranded siRNA. After that, ROG4 single-stranded siRNA complex is synthesized. Okay, so after that, RNA polymerase 5 synthesized RNA transcriptor. After that, Argo4 with a single-stranded siRNA complex can bind to the nascent RNA from polymerase 5. How about in step 2, in atypical RdDM? So at that time is also polymerase 5 can synthesize RNA transcriptor. At that time, Argo2 with a single-stranded siRNA. So this is the Argo2 with a single-stranded siRNA bind to the nascent RNA from polymerase 5. So in this complex, Argo2, single-stranded siRNA and nascent RNA complex is recruit DRM2. After that, it can induce the de novo methylation. So in this region, it's methylated. So it means that RNA polymerase 2 cannot bind to here. So it will establish transcriptional gene silencing, TGS. How about in step 3? In step 3 is the Argo4 with a single-stranded siRNA complex bind to the nascent RNA from polymerase 5. After that, Argo4 single-stranded siRNA and nascent RNA complex recruit DRM2. Okay, so and also DRM2 can do de novo methylation in the neighboring region, so resulting in DNA methylation spreading. Okay, so DRM2 can do induced DNA methylation, de novo methylation, but in the neighboring region, so resulting in DNA methylation spreading. So it will reinforce transcriptional gene silencing. Okay. So in this slide, I will explain the fourth subject, targeting of polymerase 4 and targeting of polymerase 5. So this is the canonical RdDM, which is composed of step 1 and step 2 and step 3. So at that time, during the step 1, polymerase 4 is targeted into the DNA region. And how about in step 2, polymerase 5 is targeting to the DNA region. But at that time, I did not explain how to target polymerase 4 to chromatin or how to target polymerase 5 to the chromatin. So now I will explain the details information. So initially, in step 1, SHH1 recognized H3K9 methylation. So after that, it will decode the information of methylated histone. Okay, after that, this one after that, CLSY1. So CLSY1 is ATP dependent chromatin remodeler. So maybe this one SH1 decodes the information. So maybe SHH1 direct the CLSY1 to function. What's the function? CLS1 is lose the chromatin structure. After that, so this region is loosed. Okay, after that, it will promote targeting of polymerase 4 to methylate the DNA region. Okay, so polymerase 4 can bind to the methylated region. Okay, why? So SHH1 is first recognized H3K9 methylation. After that, it decodes the information of methylated histone. So maybe SHH1 direct CLSY1 to function. What kind of function? CLSY1 has the CLSY1 is ATP dependent chromatin remodeler. So CLSY1 is the loosed chromatin structure. So after that, chromatin structure is loosed. After that, polymerase 4 is easily bind to the methylated DNA region. So this is the one mechanism. So how about the targeting of polymerase 5 to chromatin? Okay, so this is polymerase 5. How to polymerase 5 can be targeted to this region? Okay, first of all, DDR complex is involved in this step. So what is DDR complex? So DDR complex is composed of the DRD1 and DMS3 and RDM1. So DRD1 has SNF domain, so they have the ATP dependent chromatin remodeler function. So DRD1 has a loosed chromatin structure, and also DMS3. So DMS3 is they have the hinge domain. So they have SMC hinge domain. So it can interact with the DNA. So also DMS3, this interaction prevents reassociation of single-stranded DNA. Okay. How about RDM1? So this is RDM1, RDM1, RDM1. So RDM1 can bind to the single-stranded DNA. So it also reassociation of the DNA. So it means that DDR complex make the open complex here. Except that in this open complex, polymerase 5 can be targeted these open spaces. Okay, so another protein is also involved in targeting of polymerase 5 to chromatin. So we call it as SUBH2 or SUBH9. So these two proteins is involved in polymerase 5 targeting to chromatin region. Okay, previously we learned that spreads of variation 2/5. So this protein has the function, so it can recognize H3K9 methylation. So it decodes information of methylated histone. So now is that this is the SUBH, means that spreads of variation homologous to or 9. So their function is nearly similar to SU variation 2/5. Okay, so SUBH2 and SUBH9 has SRA domains. So that's why the SUBH2 can bind to the methylated CG. How about SUBH9 can bind to the methylated CHH? At that, so these proteins decodes the information of methylated DNA. At that, it promotes the targeting of the polymerase 5 to chromatin region. Okay, so this is another protein is also involved in the targeting of polymerase 5 to the chromatin. So SUBH2 and 9 has the SRA domains, so they bind to the methylated region. So this protein decodes the information of the methylated DNA. So maybe the information will be this region can be the targeting region of polymerase 5. So it promotes targeting of polymerase 5 to chromatin. Okay, so in this slide, I will explain the fifth subject, paramutation. So paramutation shows that non-Mendelian inheritance means that no dominant or no recessive. How about Mendelian inheritance? So this is the example of Mendelian inheritance. For example, genes exist in different versions called allele. For example, in this case is large R allele and small r allele. For example, large R allele encodes round sheet, but small r allele encodes the wrinkled seed. Okay. However, and large R allele is the dominant than small r allele. That's why when the genotype is large RR, their phenotype is a round shape, and also their genotype is a large R, small r. So in this case is also their phenotype is a round shape because large R is dominant than small r. Only in the small rr cases they show that the wrinkled shape. So this is the Mendelian inheritance. However, paramutation shows that non-Mendelian inheritance. So in paramutations, interaction between two alleles happens. Okay, for example, large R allele and the small r allele, between the large R, small r, interaction is happen, whereby one allele induces inheritable changes in the other allele. So this is paramutation. Once again, one allele induces inheritable changes in the other allele. Okay, so inheritable changes include the pattern of DNA methylation. So this allele induces inheritable changes in the small r allele, especially the methylation pattern or histone modification pattern. Okay, so this is the paramutation. For example, this is cone color. So this is the actual cone color. They show that purple color, but one allele induces the inheritable changes. For example, here, they showed they induces the weakly methylated. So in this case, they show that a weakly purple color. Or when one allele induces inheritable changes in the small r allele, for example, highly methylation induces highly methylation in small r allele. See, in this case, seed color will be the white is like this. Okay, so this is the paramutation. So in this slide, I will show you one example in the paramutation, especially in the cone grain color. For example, large R dash small r allele. So this allele encodes the dark purple seed, and also RST allele. So this allele encodes the purple spotted seed. Okay, and also small r allele. So this small r allele encodes the no color seeds, but in the large R dash small r is dominant than the small r, and also large R dash st is the dominant than the small r. That's why, so in these cases, large R dash small r slash small r. So in these cases, the phenotype is the dark purple seed. However, and also in these cases, large R st slash small r, because this is dominant than small r, so the phenotype is the purple spotted seed. However, there is no dominant or there is no recessive relationship between large R dash small r allele and large R st allele. So between these two alleles, there is no dominant or recessive relationship. Okay, so in this slide, I will explain the relationship between the RST allele and R slash R allele. Okay, RST allele is a function as the paramutagenic allele. and the RR allele is the function as paramutable allele. Okay, so this is the first one is that paramutable allele. We call it as RST allele. So this allele is usually the highly methylate. So this means the methylation. So highly methylated. How about another allele? So this is, we call it as the paramutable allele. Okay, so this is the R and small r allele. So when the interaction is happen, and at that RST allele induces inheritable changes in the R allele. So once again, the paramutagenic, after interaction is happen between two alleles, at that paramutagenic allele induces inheritable changes in the paramutable allele. Okay, especially here I will show you that paramutagenic allele can induce gene silencing in the paramutable allele by using the RdDM. Okay, so after paramutagenic allele induces gene silencing in the paramutable allele, so paramutable allele become to the R' prime allele. It's like this. So it means that paramutable allele is also highly methylated. So after that, R' allele becomes R' prime allele. Okay, so let me show you one example. So this cone has two alleles. So one allele is the RST, paramutagenic allele. So the other allele is the large R small r, large R small r allele. So this is the paramutable allele. Okay, so after that, the interaction happens between two alleles. Okay, so this paramutagenic allele induces inheritable changes in the large R small r allele. After that, large R small r allele is changed to large R small r prime allele. So this allele is enclosed lightly purple spotted seed. So actually the large R ST allele enclosed lightly purple spotted seed, but in these cases the large R small r is highly methylated, so they will make the large R small r prime allele. So this is the lightly purple pigmented purple spotted seed is like this. And also large R small r prime is the dominant than the small r. That's why they have this phenotype. Okay, so today we learned chapter 7. So RdDM in plants. In subject, from the subject 1 to 4, we learned the specific mechanism in the RdDM. So finally, we learned paramutation. Okay.